So now when you know that virus is depending, organism is dependent on the initial host cell, it's not capable of autonomic self-replication, it have absolutely no metabolic activity. Of course, uh, you will answer correctly on the question which culture medium to use for viral cultivation. And very often teachers trying to check your real knowledge, they can ask which agar to use for cultivation of that virus. And students starting to say that some blood agar and some other types of agar, but of course agar should not be used for cultivation of viruses because virus always needs a live cell to grow and to multiply. And in a culture medium, agar culture medium, it's a cell-free culture medium, and of course, viruses will not grow here. So you can ask how then to cultivate viruses if culture medium is not available. So in routine practice, we have three main methods of viral cultivation. One of the very first methods is inoculation in animals. Of course, you all know that animals are widely used in different fields of medical experiments and they're used for cultivation of viruses as well. We have absolutely different types of animals, monkeys, mice, guinea pig, rabbits, birds, even some chicken or similar for cultivation of viruses. And every method you will name, it always have some benefits and some penalties, and some requirements are present for every method. So if you're going to use animals for viral cultivation, of course you should know that animals should be germ free, and usually those animals are called as a SPE, specific pathogen free. Animals actually should be sterile because viral infection should not interact with any bacterial pathogen or bacterial species present as normal flora. And only specific animals can be used for cultivation of viruses. Of course, in that case, depending on viral species of type of the virus, you can identify that virus is growing in animal by death of the animal, by occur of disease or formation of visible lesions. If you know that virus is lethal for animal, then of course animal will die as the result of the infection or some specific disease will occur. And of course you all should understand that it is impossible to have virus in the composition of normal flora because virus cannot give us any benefits. They're always pathogenic, and any virus is obligate intracellular pathogen. Any contact with virus, if it is pathogenic for us, will always lead to disease. If virus is non-pathogenic for human, if it have no receptors, then it just cannot affect us. And of course, we will not have that virus in our tissues or on the surface of our skin. And presence of virus always resulting in some disease or formation of visible lesion, in some cases even death. So is it good or bad to use animals? Of course, we have some penalties of that method. First of all, any animal, it have immunity, and immunity, of course, can interfere with viral growth. And we have some viruses which can be easily eliminated by our immunity, and they cannot grow in animals because they are too weak to fight with our immune response or with animals' immune response. Of course, that method is the most expensive because you need to have specific conditions to keep animals. You need a lot of space. Animals are slowly growing in the contrast with some cell or the same bacteria. And of course, you need a lot of time to grow the animal, to take care of animal. You need to feed animals. You need to provide them with specific requirements. Of course, it all takes money. You need the staff to take care of animals, and animals always have it is always a risk to handlers because animals can bite, they can damage us. Of course, it is always some risk of trauma, or if incorrect handled, animals can be ill with some diseases which are dangerous for us as well. And we said that we have some viruses which are specific only for one or few animal species, and we have very narrow viruses which are pathogenic for humans, but absolutely not pathogenic for animals. And of course, in that case, we cannot use animals for cultivation because they will not be infected. Receptors specific for virus are absent in those animals. And of course, the main problem with that method is animal welfare. And we have a lot of protesters and a lot of organizations who are trying to save and prevent animals from that unpleasant death. And we have, in some countries, we have a lot of problems, and some types of animals, for example, are prohibited for studies. And of course, it is always a problem. It is a doubt because animals are actually alive. Some of them are clever. They will understand what how they are used for and what is happening to them. And of course, it is always internal problem because to kill animal, it's not always so easy. But that method, of course, have some benefits. First of all, animal models, especially if it is some close to human animals, they are very close by pathogenesis, by clinical symptoms, and we can study not only actually we can use animals not only for cultivation of viruses, we can use them as a model for different treatment options, for testing of different diagnostic tools, vaccines, for example, or some other. And of course, diseases, duration of disease in human and animal very often are similar. Second benefit, you have all required tissues in one place. And according to type of inoculation, you can have infection of liver, infection of brains, infection of muscles. Any tissue which is present in human can be identified or can be found in animals as well. And we have the same methods of infection. Animals can be infected intranasal, for example, by the use of some aerosols. They can be infected through food consumption or um, subcutaneous infection. So again, model of infection is the same as in humans. And of course, if we need to evaluate, for example, immune response on viral infection, then animals are perfect model because immunity mechanisms of immunity and response of immunity in animals is absolutely the same as in humans. And if we need to study, for example, oncogenic properties of the virus, is it possible that some cancer is caused by virus and originate initially from viral infection? Then animals are the perfect model. We can study rate of cancer in infected animals and virus-free animals, and actually it is the only possible model for oncogenesis studies. But again, due to multiple penalties, that method is limited, and usually model laboratories trying to use other models if it is possible, and number of animals used for different medical experiments is decreasing with the time. So another type of virus cultivation, more simple and more available, it is use of chick embryo or embryonated eggs. Of course, eggs again should be marked or labeled as a SPE, means they should be free of germs and free of pathogens. And usually those eggs are arrived from SPE birds. So birds should be germ free, and of course, in that way, eggs will be germ free as well. So, embryonated eggs are the most suitable for primary isolation for identification of viruses. Eggs are used for reproduction of some viruses to obtain vaccines, and again, infection, viral infection in chick embryo can be identified by death of embryo, formation of different deformities, hemorrhages, edema of tissues, and formation of some specific intracytoplasmic inclusions or inclusion bodies in the tissues of chick embryo. They can be identified with the help of microscope or other techniques. As well, chick embryo have different tissues, and according to type of inoculation, you can have access to different tissues. It is shown in that picture in that slide that we can have inoculation in some yolk sac, amniotic inoculation, allantoic or other. But that method have two main penalties. First of all, tissues of human are very different from tissues of chick embryo, and only few human viruses can be grown in chick embryo. So of course, it's that method is very restricted, and we can use it only in a few cases, and main number of human viruses cannot be cultivated in chick embryo. And different site of inoculation should be used for different viruses. So it's not just you can infect a chick embryo and that's all. You should know which exact position of egg should be used for inoculation of precise virus. And of course, the technique is difficult because you will not see internal structure of the egg. You can see only that shell, and it's not transparent. You cannot see embryo, and it is only some markers or labels how to infect that chick embryo with the help of the virus. But of course, you always have a chance that you will inoculate virus not in correct place, and in that case, your virus will not grow. And of course, you will not see viral growth. You will not see formation of those lesions, hemorrhages, or other. If you will see that chick embryo is absolutely intact, means that inoculation of virus was performed incorrectly. But we have some benefits of that method. Of course, maintenance of that method is much easier than inoculation of animals. Less labor is needed, and of course, that method is much more cheap because you don't need to feed chick embryo, you don't need to feed embryonic egg, you don't need to grow it. Of course, birds producing much more number of eggs than animals producing generations, and that method is faster. Of course, embryonated eggs are readily available, and you don't need to have some specific permission for cultivation for maintenance of animals because it is really required. And laboratory, if it want to deal with animals, should have specific license, should have permission from government. In the case with eggs, it's much more easy. You just can buy that egg in any fabric, any farm, and use it. Again, still, if it is even if it is limited, but we have wide range of different tissues and fluid in one place, and it is possible to cultivate multiple types of viruses. And chick embryo is always free from contaminating bacteria. It have no immunity, and immune system is absent in chick embryo, and you will not have interaction with the immune system, with the immune cells, and it is always easier to cultivate viruses in chick embryo. But again, in human medicine and human practice, that method is limited mainly due to very different structure of tissues in human and chick embryo. And the most popular and the most perfect in nowadays method of viral cultivation, it is use of cell tissue cultures. So actually, method is some kind of similar to cultivation of bacteria, but you should have culture containing cells. It's not agar culture. It is specific type of culture used to support the life of eukaryotic cells. And here we have several types of cell cultures. First, the most simple, the most easy is a primary cell culture. So to obtain primary cell culture, usually you need to take some amount of normal cells from fresh organs. It is type of biopsy or maybe some material left from surgery when some organs are cut or some tissues are cut, and you can use that tissue. You need to put it in specific medium to support the life of the cells, and you will have the same cells as present in organ, but already in that culture. Of course, you will have very limited growth, and usually cells obtained from primary cell culture have only few cycles of replication, and duration of life is limited. So primary cell cultures are used for isolation of viruses to prepare vaccine or some primitive experiments. And mainly we use human amnion cell culture or rhesus monkey kidney cell cultures. So they are more suitable for some experiments. But again, only a few generations those cells will give. After that, those cells will die. And usually primary cell cultures are used only for single experiments. So to obtain more suitable culture, we have diploid cell cultures. It is a semi-continuous cell cultures, and they are derived from initial primary cell cultures. Again, you need to take that biopsy of organ or some tissues from organ, then by multiple mixing and usually addition of some trypsin or other compounds, you need to dissolve primary cell culture and break them into multiple single cells. And those cells of single type, they can be used for reinoculation and further cultures. And those cells, they still will have diploid chromosome number. They're capable for about 50 serial subcultures. And of course, it is only one specific type of the cell. They all are kind of clones, and diploid cell culture can be used for isolation of fastidious viruses, and again, usually it is used for production of vaccines. So we have again multiple different types of cultures, but one of the most known is WI38. It is a culture of human embryonic lung, and we have rhesus embryo cell strain, which is called HL8. Two main examples of diploid cultures used. And the most perfect type of cell tissue culture, it is continuous cell cultures. It is cell of a single type derived usually from cancer cell. And of course, you all know what is cancer. It is phenomenon when cell can lose control over multiplication. It will multiply continuously, unstoppable. And of course, cultures which are obtained with the help of cancer cells, they will have continuous serial cultivations. They are growing faster, and the main difference with previously mentioned cell tissues cultures, they have haploid chromosome number. It is always haploid number, and cells are divided by asexual reproduction. And of course, those cells have unlimited number of multiplication. That's why they are always called permanent cell lines. So every cell in that cell line is a clone of maternal cell. So all cells are absolutely identical. Here we have HELA or HELA strain, which is derived from the cervical cancer of human. The cell line is the most known. It is used for isolation of viruses. And of course, that type of tissues is the most suitable because actually you can use it again, and again. They always multiply and continuously, and it is easy to handle those cells because they are less fastidious. It is a cancer cell. It just multiplying and always giving you new generations of further cells. So when we discuss culture medium, I already showed you those flasks, and usually we have specific type of laboratory dishes for cultivation of viruses to maintain cell lines. And again, it can be flask, but you can have, as you can see, when petri dishes with culture for cells, but it is always liquid, and mixture of cells are growing here. You can put it in simple incubator, and again, you will have growth of those cells. So by principle of use, it is very similar to agar culture medium, but of course, for viruses, we have specific cultures. As you can see in that slide, it is a photo of culture lines obtained with the help of electron microscopes. Every culture of cell is absolutely identical. You can see that all cells are absolutely identical, and according to different types of tissue, we can have cell lines for specific organs. And of course, it is easier for us because you can obtain any tissue you need. You can take cancer of muscle tissue, you can take cells from brain cancer, and you can obtain any cell line you need. If you need to cultivate neural virus, take cancer cell from neural tissue and you will have that line. Of course, that method have as well some benefits, some benefits and some penalties, and the most important penalties of that method is a requirement of a specific good laboratory facility, and you need to have opportunity to obtain, to purchase that cultures for cell lines. Of course, you need specific devices, specific incubators. You just have laboratory which is absolutely free of some bacteria or concomitant microorganisms. It is usually more expensive. Culture is more expensive, and maintenance of that equipment is more expensive. And all common things are more expensive to support cell tissues. Of course, you always need specific trained technicians, and laboratory staff should be trained and licensed for work with the viruses. Of course, again, you need to understand that viruses are mainly pathogenic for humans. They can infect us, and it is more strict requirements for general safety measures. But as we said, that method have more benefits than penalties. Of course, you can grow almost every virus with the help of cell tissues. If you need some specific tissue, you can just obtain that cell culture, and use it. You can grow viruses in bulk, and actually cells can be stored for longer time period. Of course, it's much easier to support life of cells than to support life of monkey or some other animal. And method is relatively easy. So it is just some petri dish or flask with the culture, just perform some simple activities, and you will have result. You don't need to have some specific method of inoculation. You don't need to evaluate some additional symptoms. It is just cell tissue which is always suitable for growth of one precise virus. And that method, of course, is highly sensitive because you always will use required tissue. And of course, that method have absolutely no ethic problems because those cell lines, they are kind of not alive. It's not an animal. You don't need to kill anyone. It is usually cancer cell. No one cares about the health of that cell. Of course, it's absolutely easier for laboratory staff to deal with that method because you don't need to kill some alive beings, and of course, it's much more easier. So it is the common idea of how to cultivate viruses, and one more problem, and actually some students even asking, and not only students, doctors asking, who are really interested in trying to understand some common problems. They asking how it is possible that viruses actually appear, and what is the origin of the virus? What is it, some part of initial cell, or actually how is it possible that some infectious particle, which is absolutely inactive, exists in our planet, and moreover it can infect and cause serious damages in our tissues? And it is again a common doubt because in nowadays we cannot prove how viruses appear. And we have only several theories, some suspicions based on some evidence how virus could appear on our planet some centuries, millenniums ago. And I will talk about three most common theories. They have some uh, good basis and they have some explanation. First theory, it is a regressive theory. It suggests that viruses initially were some unique cellular structures. Maybe they were cellular parasites, and initially they had complex structure, maybe similar to bacteria, but there were complete independent particles. But with the time, they lose one by one, they lose some excessive genes coding for structures which are not obligate for surveillance. For example, cell wall was lost because actually it's not important for viruses. Some cytoplasm was lost because it's again not important in genetic parasitisms, and actually it is only factor which make the cells more sensitive to environment. And with the time, one by one, different organelles and structures was lost, and viruses transformed, they regress to those agents we know now. And the theory initially is based on two microorganisms we'll discuss in the further part of the lecture, chlamydia and rickettsia, because actually there are some intermediate form of life between bacteria and virus, and maybe it is a time period when bacteria regress into virus. And chlamydia and rickettsia can serve as an example of regression of bacteria to some more primitive form of organism. Uh, but again, the theory has some penalty. For example, it cannot explain why current parasites, known parasites, for example, the same chlamydia and rickettsia, are still so different than viruses, and structure is absolutely different. And of course, it's not really possible to explain why some complex structures regress to so primitive. And anyway, the structure of viruses slightly, even significantly different than structure of bacteria. It's absolutely different. And of course, that theory is not still is not common in medical and scientific community because it's weak theory, but it is still have some base. And again, the chlamydia rickettsia can act as an example for regression of microbes to some primitive forms of life. So second theory of viral origin, it is a cellular theory, and it suggests that viruses, current organisms we know as a viruses, it is a result of splitting of genome of cells, because actually the only thing which is the same in viruses in our cells, it is a genome, and maybe that splitted genetic material evolve to self-replicating forms. And again, that theory is based on transposons and plasmids. Of course, you all remember from biology that we have independent locuses of genes, jumping genes they are called, which are kind of still part of our cell, but they're independent. They can jump over different locations, they can insert into the one place, they can be rejected and jump to other place. And maybe in some time period of evolution, those genes were separated completely of our cells and became independent. And that theory seems like a good theory, but again, it cannot explain how capsid appeared. Because in our genes, of course, we have no information about structure of viral capsid and other structures such as glycoprotein spikes, how they appear, because actually it is absolutely unique gene which cannot appear from anywhere. And again, it is a penalty why, and it is the reason why the cellular theory is not very common in community, but again, maybe some part of the theory is true. The last theory, the third theory we'll talk today, is the theory of co-evolution, and the theory seems like a most confirmed by different facts and by our biology. The theory suggested initially, from the very beginning, when life only appeared in our planet, viruses already appeared as a separate form of life. It is a result of combination of different complex proteins, nucleic acids, and it just happens so that those components combine together in one organism, and viruses appeared from the very beginning in the same way we know them now. So the theory suggests that bacteria appeared. Actually, we cannot understand and we cannot explain how bacteria appeared, how some fungi appeared. We know just they appeared and that's all. Maybe viruses is the form of life which appeared together with bacteria and other forms of life, and initially they were independent and they were parasites from the very beginning. But again, that theory cannot really explain how dependent form of life could appear independently. So if virus was dependent on eukaryotic cell, how can it appear independently before eukaryotic cell appeared? Or maybe together with eukaryotes, but already. So again, any theory have some points we cannot explain. But again, we cannot explain actually appearance of animals of so large types of animal species. We cannot explain appearance of bacteria and other. So only theories, and still we have some suggestion. If you will have your own theory, maybe you will write a paper into some scientific journal, and you will provide your own opinion. It will be fourth common theory in current slide, but still nowadays we have three more, most common theories. And of course, nowadays, you know that we have multiple different options of genomic studies, and current studies show that viral genes have absolutely no homology with genes of bacteria, with genes of archaea, or with genes of eukaryote. And initially, development of virus was absolutely independent, and it had very distant branching. Means that viruses initially, it seems like viruses initially really were independent form of life. It's not a part of some bacteria. It's not a part of our transposons. Initially, structure of genes were absolutely different. And again, it seems like the third theory of coevolution is the most acknowledged nowadays. So we said already about chlamydia rickettsiae, and we mentioned that viruses, we have a theory that viruses can be a result of regression of bacteria. So let's say about a few words about rickettsiae and chlamydia. So we have two unique bacteria which are very different from other bacteria. And the main difference is that bacteria, similar to viruses, uh, depends on initial host cell. And chlamydia is obligate intracellular bacteria. It cannot multiply independently. And the main reason of that, chlamydia lack several metabolic and biosynthetic pathways, and it cannot produce energy itself. So it depends on host cell for intermediates such as ATP. And that's why bacteria cannot multiply itself. It cannot just produce energy, but it has unique apparatus to steal energy from the host cell, eukaryotic cells. And if chlamydia will get inside eukaryotes, it can consume energy produced by a host cell. And that way, chlamydia is not really a bacteria, because when we describe in bacteria, we're saying that it is form of life capable for autonomic independent self-replication. But chlamydia is not that bacteria. That's why very often in literature, Chlamydia rickettsiae are named as atypical bacteria. It means that it's not typical one. It has some important differences. And the difference is because bacteria depends on the eukaryotes. So in common, chlamydia still belong to bacteria because it have the same structure, the same organelles. It is prokaryotic in composition, and actually all properties of bacteria are applied for chlamydia as well. Then it can be stained by gram-it gram-negative. It's non-motile and non-sporin, Coccobacillus in shape. And the main difference is that bacteria is difficult to find in environment because it can survive, it can multiply, and can have metabolic activity only within eukaryotic cell. So for identification of chlamydia, usually gram stain is not used. We're using some techniques suitable for identification of intracellular parasites or staining of eukaryotes, such as the GIMS, the Romanovsky, Jimenez, or other techniques. And similar to virus, chlamydia exists in two forms. It is extracellular form and intracellular form. So if we said that we have virus in virion, in the case of chlamydia, we have elementary body and we have reticulate body. So if you will identify a bacteria outside the host cell, it is extracellular form. It will be infectious. It can infect suitable cell or suitable tissue. It's non-replicating because it has no energy. It cannot multiply. It is very rigid and much smaller in size. It is elementary body. So again, similar to viruses, chlamydia have affinity only to certain tissues, and chlamydia is specific for epithelium. It is microbe which can infect only epithelium of different tracts, epithelium of respiratory tract, of urogenital tract, but it is always found in epithelium. And again, it's making that bacteria more close to viruses than to bacteria. And when chlamydia will recognize suitable tissue, suitable cell, it will invade inside the host cell, and here in cytoplasm, it will be transformed to non-infectious but active reproductive form. It will be less rigid but much bigger in size. And that form is called reticulate body. It is non-infectious intracellular form. Again, it will have access to energy inside the host cell, and it can reproduce, it can perform reparation processes, it will have metabolic activity. And similar again to viruses, that microbe exists into two forms. And it is again making that bacteria atypical because usually any other bacteria is the same in any environment. Because microorganism is obligate intracellular parasite, it cannot exist for too long time period outside the host. And usually that disease, that bacteria or diseases caused by that bacteria are transmitted by direct human-to-human contact. It can be air, sexual, or other types of contacts, but always it is contact human-to-human. And of course, we can identify live and multiplying bacteria, active bacteria, only within human cells. Outside the human, it cannot reproduce, it cannot maintain a population of microorganisms. So here on that picture, you can see, of course, reticulate body. It is located inside eukaryotic cell. You can see that large, dark purple cells, it is a eukaryotic cell, and within it, you can see spherical light balls. You can see it marked with the green arrows. It is intracellular chlamydial cell. It is grown in cell cultures. And again, it is a property making that bacteria more close to viruses. You cannot cultivate chlamydia in agar culture because it is required cells. And for cultivation of rickettsia and chlamydia, again, we need a cell culture, so we can use animals or some other forms of cultivation, but it is never agar. So, similar to viruses, intracellular bacteria, for example, chlamydia, have unique type of reproduction, and of course, cultivation of bacteria and infection of that bacteria is more complex initially in environment by human-to-human transmission. You will have always elementary body identified. It is the infectious particle transmitted from one person to another. And when elementary body will find suitable host cell, it will attach to a surface, it will invaginate within the cell, usually by endocytosis, and it will enter the host cell, and here it will start to grow and enlarge in size. In environment, bacteria need to resist aggressive conditions of air, temperature, moisture, sunlight, and bacteria will be smaller in size. It have no energy, it cannot maintain large forms, but it will be more rigid. Cell wall is more rigid, it's bigger and thicker, and bacteria is more resistant to environmental factors. But now, when bacteria enter the host cell, of course, no sunlight, temperature is stable, a lot of nutrients around, and energy. And bacteria will grow. Initially, it will enlarge in size, become oval or more like a rod shape. It will be less rigid in the structure of cell wall, and bacteria will start activity. Now it have access to energy, it will be metabolically active. And that form is called as reticulate body. And of course, the main task of bacteria is to grow, to grow, to grow in size. And when size of bacteria will reach maximal possible, usually it occurs within some 12 to 20 hours. Bacteria will split in two, and every new form, any new microbe, will initially appear as elementary body. So one reticulate body is approximately twice bigger in size than reticulate body. And during growth, one reticulate body will split into two elementary bodies. And again, bacteria is multiplying by binary fission, but only within eukaryotic cells. And further multiplication will occur in geometric progression, and every new daughter cell will give two more further cells, and bacteria will multiply. So in some time point, you can identify host cell and presence of both elementary bodies and reticulate bodies in one cell according to stage of bacterial reproduction. And bacteria will multiply as long as some enough space in the host cell will be available for further multiplications. And of course, when host cell will be limited, all space will be occupied by bacteria. Cell will be broken, it will just destroyed, and a lot of elementary bodies will be excreted outside, and those cells can affect new target cells, or they can be released in the environment and infect other hosts, other humans. And that life cycle again is very similar to viruses, and making that bacteria really atypical. Second microorganism, which is again very similar, it is a bacteria which is called rickettsia, and in that picture you can see rickettsia in the host cell. Again, that huge pale pink in color, it is eukaryotic cell with dark violet nucleus, and red long elongated rod bacteria, it is rod-shaped red bacteria, it is rickettsia. Rickettsia, similar to chlamydia, is atypical bacteria. It have almost the same properties as chlamydia. It is depending on the host cell. It have as well two forms of life, and similar to chlamydia, extracellular and intracellular. And as well, rickettsia can be identified as bacterial or by all properties. It is gram-negative, non-motile, non-sporing, rod-shaped bacteria. But again, according to different types of or different forms, it can have different properties. So rickettsia exists in vegetative form and silent form. Silent form, it is an analog of elementary body of chlamydia, and vegetative form is an analog of reticulate body. So silent form, it is extracellular form, usually smaller in size with more rigid cell, and vegetative form, it is the active multiplying and reproducing form. So in the contrast with chlamydia, rickettsia depends on endothelium, means it can affect only blood vessels. And of course, to enter blood vessels, bacteria need some vector. And usually rickettsia is transmitted by arthropods, different types of arthropods, ticks, lice, fleas, mice, chiggers, and other. And that's why diseases caused by rickettsia are mainly zoonotic, means they're transmitted from animal to humans by blood-sucking arthropods, but as well we have some diseases caused human-to-human transmission. And rickettsia, again, similar to viruses, have affinity to endothelial cells that can affect only one tissue within human bodies. So for cultivation of rickettsia as well, you cannot use any agar, and usually uh, we can use animals, so in some cases we can use even arthropods, for example, ticks, for cultivation of rickettsia if it is possible to have transovarian transmission of that microorganism. So those two unique forms of life, rickettsia and chlamydia, it is a perfect example of some intermediate existence where it is not a bacteria anymore, but it's still not a virus. And there are two types of bacteria. They are very unique. Again, they're very often called atypical, and detailed properties will be discussed in next semester when we will have private microbiology. So one more question, and actually again, it is unique form of life, and in Paniker book and a main number of different student books, that question is not very good described. So I will try to make it more clear for you and more easier for you to understand what is it. So sometimes those forms they are described as a separate form of life, but actually it's more correct to name them as bacteria with modified cell wall. So we have several types of modified bacteria. First one is a protoplast. And actually in your book it is very good written how to obtain protoplast, which chemicals to use and how long to treat with different chemicals, how to obtain, but not that is important. You should just understand what is protoplast, what is some spheroplast, L-form, and other. So if you talk about protoplast, again, initially it is a bacteria, but it is a bacteria which lost cell wall by some reason. So protoplast, it is bacterial cell without cell wall. And actually the whole other structure will be absolutely the same. Protoplasts have the same organelles. They are covered with plasma membrane, but because cell wall is absent, plasma membrane cannot maintain the shape of that bacteria. And usually you can see cells, they are presented in pictures, in both pictures on that slide. It is simple light microscope photo. It's not electron, it is a light microscope. And you can see that it is some spherical things which are present in high amount. They can differ in size. Actually, they can change the shape according to environmental factors. If something will press on that bacteria, it will change the shape because nothing, no organelles to maintain the shape. And usually protoplasts are identified when gram-positive bacteria lose the cell wall. And actually you can say that protoplast, it is gram-positive bacterial cell which lost cell wall. Reasons can be different, but the main idea that it is a cell which will not be susceptible, for example, for some antibiotics, because penicillin, for example, is acting on the cell wall. No cell wall, no activity of antibiotics. That microbe will be more sensitive to environmental factors because they lose natural protection as the cell wall. And it is a phenomenon which can be observed in medical practice after therapy of patient with penicillin, or the same forms can be obtained in laboratory experiments if they are required. But by common properties, these simple bacteria are very active, alive, and have the same properties. The second form of modified bacteria, it is spheroplast. Again, it is a bacterial cell, initial bacterial cell with fragments of cell wall. If protoplast is the cell without cell wall, complete absence of cell wall. Spheroplast, it is a bacterial cell with fragments of cell wall. Usually that phenomenon is identified in gram negatives. So you can say that it is gram negative bacteria with partially destroyed cell wall or with only fragments of the cell wall. Again, that microbe will lose natural organelle required to maintain the shape. And spheroplasts are usually much bigger than initial cells. They cannot hold that pressure which is present in initial cell. And cell will enlarge, it will extend, and usually they can occur as some oval or spherical particles without fixed shape again. If something will press bacteria from a side, it will become flat. And by all properties, it is still bacterial cell. It will have the same organelles, internal structures intact, and only cell wall is partially destroyed. So it is possible, but very rare phenomenon when spheroplast can regress to normal cell, and cell wall can be built to a common manner, and bacteria will again appear as a normal one, but that one is really rare. So the main property of protoplast and spheroplast both, they can usually they will die. They became more sensitive to our immunity to environmental factors, and usually they die. But sometimes it is possible to have transformation to stable ale forms. And ale form of bacteria, it is a stable form of bacteria with partial or complete absence of cell wall. So again, it is similar to proto and spheroplast, but usually both are unstable, and it is some time period when normal cells lose cell wall, and after some time period they will be destroyed and they will die. So it is very short time of life between dead bacteria and normal bacteria, due to again mainly some chemical factors. You gave antibiotic, it destroyed cell wall, bacteria can exist for some time period in that spheroplast or protoplast shape, and then it will die by other reasons. But sometimes we can have progression to L forms, and L form is a stable form of bacteria with partial or complete absence of cell wall, and that L form is able and capable for further development. Again, internal structure is intact and organelles are absolutely the same. An L form can exist for long time period. It can multiply and give a new generation of L forms. And in unfavorable conditions, that L form can progress to non-stable L form. And that L form, of course, will die very easy. And rarely, but we can have transformation of L form back to normal condition, to normal state, if that unfavorable factor will be removed. For example, you treated subject with penicillin, cell wall was lost, bacteria transformed to L form. You stop therapy, penicillin is removed, bacteria will again synthesize that lost cell wall and turn back to normal condition. So in the case of L form multiplication, of course, binary fission is impossible because cell wall is absent, and L forms are multiplying by budding, similar to some candida fungus. They can produce buds and new cells will appear. But in general properties, it is always the same form of bacteria, but with some partially or completely destroyed cell wall. Uh, as well, we need to say a few words about third atypical microorganism, which as well acting as a parasite, it is mycoplasma. So mycoplasma, it is bacteria which naturally lacks cell wall. And by nature of bacteria, cell wall is absent. It's not a type of protoplast or spheroplast, it is separate family of microorganisms. And by nature, that microbes have no cell wall. So, of course, it will have a very similar appearance with the protoplast or spheroplast. It is very unstable in form microorganism. Nature it have a spherical form, but due to environmental factors, it can be pressed or and have some oval shape or other shapes. And that bacteria depends on human and animals, because to remain alive, microbes have tri-layer of plasma membrane made of complex sterols, and those sterols cannot be produced by bacterial cell. It is always should be stealed from eukaryotes, and mycoplasma is act as a surface parasite. It will stick to the surface of host cell, and it will consume sterols from mainly human and animal cells. So bacteria can survive in an environment, and usually that microbe is transmitted by air or through sexual contact, similar to chlamydia. And mycoplasma is widely found in environment. It is capable for surveillance outside the host, but multiplication and growth is possible only when sterols are found in environmental conditions. And we have some species of mycoplasma which can be grown in cell-free culture medium and agar medium, but it is rare exception. Usually, again, a live cell is required to maintain or to support the life of mycoplasma. So just few words about more even primitive forms of life. So we said that we have virus which is looking like the most primitive, but actually uh, we have a type of microorganism which is called viroid. It's not even a virus, it is viroid. So viroids are subviral particles. They're even more primitive in structure. Usually they are presented by naked RNA molecules without any coverage. It is simple molecule of RNA. And viroids, of course, are too primitive. They are not identified in humans, but they are very important plant pathogens. And genome of viroids holds no information about some proteins or other compounds, but they can they can interact with a host cell, and that RNA can begin integrated into the host cell and used for self-replication. So, of course, those viroids are more sensitive to environmental factors, and they cannot infect humans and animals. They can be easily deactivated by our immunity. But in plants, those organisms are circulating and causing some diseases. So in that photo, it is electron photo. You have two types of viroids, linear viroid and circular viroid. They are marked with an arrow, so you can see that it is very small molecule. It is single molecule of RNA. And one more, even more primitive in structure. Again, we cannot call it even microorganism, but it's still prion. Prions, maybe you heard something and you know. It is infective protein. Actually, it is simple protein. It have no even RNA or DNA. It is just a protein molecule. It's more primitive in structure than viroids. And those proteins, they can interact with the host tissues, and they are able to initiate production of self copies. And actually, in our tissues, in animal and human tissues, we have already prion proteins, and naturally prion proteins exist in two isoforms. It is normal form or PRPC. It is shown in the left part of the slide. And we have altered form of that protein or prion, PSC. So it is modified protein. And normal prion proteins, they are widely found in animal and human tissues. Usually they are used as a cell surface glycoproteins, and in highest levels they are identified in neurons of brains and spinal cord. It is a normal structure which have certain functions in our bodies. And normal proteins, they are, of course, widely found in our tissues. They perform in normal functions and actually neurological activities possible only due to presence of normal prion proteins. But uh, they may exert cytoprotective activity against some internal or environmental stress factors. Um, they can initiate apoptotic programs, and the main problem is that normal prion have specific and certain polarity and chemical charge, and it have unique structures. You can see in the picture it is spiral structure and located in certain um geometry that protein will have normal functions. But when abnormal or diseased or altered prion protein will get very close to normal one, it will change three-dimensional structure of normal protein, and that normal protein will be transformed into prion isoform. So actually, we cannot name it as a reproduction because actually it's just modification of normal protein to abnormal one. But abnormal protein cannot perform normal functions. And usually very shortly it will lead to a death of cell that protein is attached to. And the main problem of prion diseases, if and usually prion diseases are transmitted by consumption of some uh, food which have that abnormal protein. Uh, we cannot identify that protein by any usual method because we cannot cultivate proteins. It's not active. It cannot multiply. It cannot cause any damage to visible damage to cells. It's just modification of three-dimensional structure when two proteins are coming into close contacts. And that is a problem because again it is simple protein. It have no some specific receptors. It have no activity. It's very difficult to identify that protein in food. And we have different again proteins in humans, in bovines, and usually the structure is very similar. If you take a look, if you take a fast look at that picture, you will not identify significant differences between human and bovine proteins. That's why prions have no host specificity. They don't care about animal or human tissue. They can affect both. And we will not talk a lot about diseases. If you're interested, you can read in some internet or other books. But usually human diseases were initially animal diseases, and in animals prions can cause so famous diseases as scrapie in sheep or chronic wasting disease in deer, bovine spongiform encephalopathy, or common name is the mad cow disease. And usually those diseases are limited to neural tissue, and of course they will change behavior of animals, behavior of humans. And human usually became infected by eating not properly cooked or even fresh meat products of animals, especially if it is a brain tissue. And of course, proteins can be inactivated by proper cooking, by heating, and usually it is improperly cooked or fresh meat products. And we have a range of diseases in human, for example, Creutzfeldt-Jakob's disease or um, fatal familial insomnia, Kuru disease, and or laughing disease, and some other. And actually, in that picture, it is Kuru disease shown in the upper picture, when, or it is as well called laughing disease, when people will lose highest mental abilities. People always smiling, laughing. They became not very clever, and mainly all prion diseases are fatal diseases because uh, constant destruction of neural tissue will lead to death and, of course, lethal outcome. One more problem of prion diseases, we have no treatment for those diseases because actually, again, protein is the same as we have in our own tissues. And if you try to destroy abnormal prime proteins, we can destroy our own as well. And any disease till nowadays is incurable, but it is easily prevented by proper cooking, by consumption of checked food. So if you'll again try to study some information about those diseases are limited to some native populations. Again, in that picture, it is native population. It is aborigine of some countries who have natural religions or cults of eating of fresh meat. And some diseases are observed only in cannibals religions when human flesh is used for food profit. And of course, those diseases were more prevalent when more aborigines were identified in different countries. Nowadays, of course, cannibalism is prohibited, and human diseases are very rarely observed nowadays. But transmission from animals to humans again is possible. So if you will take a look, for example, in CT scan of brains, in left part it is normal brain tissue, and right part it is affected brain tissue. You can see that amount of brain tissue is decreased significantly. Structure is deformed, and usually prime diseases manifest with neural degradation, with degradation of different functions, and people slowly will become unintellectual. They will lose common highest functions, and with the time, massive destruction of neural tissue will lead to involvement of vital sites such as breathing site or some BP site, and people, of course, will die because of inability to perform common vital functions. So if you will take a biopsy of those tissues, that's why actually diseases were called spongiform, because in biopsy of brain tissue, you will have formation of vacuoles or vesicles, which will replace normal neural tissue. And again, you can see that in lowest picture, in right picture, brain tissue is really looking like a sponge. A lot of cavities, a lot of bubbles inside, and of course, the significant decrease of normal tissue will lead to loss of multiple neural functions. So, if you're wondering about those diseases in more details, then again, you can search, you can find a lot of articles, and some diseases are really interesting. And again, the main idea, we cannot identify them properly, we cannot diagnose disease properly, and we cannot treat diseases. So prevention, again, is only non-specific, and the main option to avoid that disease is proper cooking, or not taking in food some fresh or undercooked, especially brain